Cancer is the most common cause of death in Japan, and cancer mortality rates continue to increase. Cancer diagnosis can be devastating. However, over the past decade, the 10-year survival rate has been steadily increasing. Recent advances in cancer genome profiling offer patients the hope of receiving personalized therapy based on their cancer's specific genetic makeup. Precision medicine is based on the observation that some patients respond better than others to a specific treatment, and that this difference might be due to differences in which genes are affected. Therapies are then designed to specifically target the affected genes and pathways, resulting in more effective treatments with fewer side effects. In this video, we will discuss how cancer genome profiling can help a physician determine whether a patient might be a good candidate for targeted therapy. Complete genomes can now be sequenced in just a few hours, but the output is in the form of millions of short reads. In order to make sense of this jumble of data, we need a map. And for this, we use the Human Genome Reference Sequence. Currently on its 38th version, the Human Reference Genome does not reflect the genome of any specific person, but rather is an amalgamation of multiple anonymous individuals. The sequencing of the human genome cost $3 billion and took an international team of 20 research centers 13 years to complete. It stands as one of the greatest collaborative scientific achievements in history. With no existing genome sequence to refer to, the International Human Genome Sequencing Consortium, or IHGSC, had to start from scratch. First, the IHGSC built a linkage map. During meiosis, the maternal and paternal copies of each chromosome line up, and some regions are exchanged in the process of recombination. Genes that are close to each other on a chromosome tend not to recombine as often as genes that are farther apart, and genes on separate chromosomes do not undergo recombination at all. By examining recombination rates among genes, the researchers were able to determine which genes were on the same chromosomes and roughly how far apart they are. Physical map. The linkage map revealed the orientation of genes, but not their actual location. To create a physical map of the genome, the researchers collected blood samples from 20 male and 20 female donors and selected 2 samples of each. And then they removed the labels. That means that even to this day, neither the researchers nor the participants themselves know whose DNA is actually represented in the reference sequence. Having said that, it is known that much of the work was done using DNA from a male donor from Buffalo, New York, codenamed RP11. Next, DNA was extracted from white blood cells from the selected samples. Because no single laboratory could sequence more than a small part of the human genome's 3 billion bases, the DNA was fragmented into smaller pieces of about 150,000 base pairs and amplified in bacteria. BAC preparation. The bacterial artificial chromosome, or BAC, was created from each fragment as a way to store and copy the fragment. In this method, a fragment is inserted into a plasmid, which is taken in and copied by the bacteria and passed on each time the bacteria divides. Each clone was then fingerprinted by digesting the plasmid with a set of restriction enzymes, which are enzymes that cut DNA only at specific sequences. When run through an electrophoresis gel, the sizes of the resulting pieces create a unique pattern. Since the BAC patterns overlapped, the researchers were able to sort the BAC fingerprints into genome order. From this BAC library, the IHGSC selected a set of about 20,000 overlapping BACs and assigned them to individual research centers for sequencing. The 150,000 nucleotide sequence in a BAC was still far too long to sequence directly, and was further fragmented into subclones of about 2,000 nucleotides each. Finally, the subclones were sequenced using Sanger sequencing. In this method, DNA is separated into single strands, and a primer is attached at one end. DNA polymerase is then added to create a phosphodiester bond between the 3' hydroxyl group of the primer and a deoxynucleotide triphosphate, or DNTP, complementary to the template strand. DNA polymerase continues to extend the chain by adding additional DNTPs to the 3' hydroxyl group of the previous base. However, a small number of modified dideoxynucleotide triphosphates, or DDNTPs, are also added to the mixture. These modified nucleotides lack a 3' hydroxyl group. When one of these bases is added to the chain, polymerase is no longer able to attach additional DNTPs, and the chain terminates. However, because the modified bases are rare, other chains are able to extend a little further before encountering a DDNTP, and others extend farther still. Eventually, a DDNTP is inserted at each possible position along the template. When run through an electrophoresis gel, the resulting chains migrate based on their length, sorting themselves into the original order. Each modified nucleotide also contains a fluorescent label, with a different color for each nucleotide type. The sequence of colors is read, and the original nucleotide sequence is reconstructed. Assembly and gap closing. Once sequencing of each subclone was completed, the individual sequences were stitched together like an enormous puzzle, and finally assembled into complete chromosome sequences. However, many gaps remain in the completed sequence, and no technology at the time could sequence reads long enough to bridge the heterochromatin gaps. In fact, the telomere-to-telomere sequence was not finally finished until 2021, with the use of the long-read technology from Pacific Biosciences and Oxford Nanopore. FASTA files. What does the human reference sequence look like? We mostly work with the genome data in FASTA format. This is a simple text format in which the first line starts with a greater than sign, followed by the name of the sequence and a short description. The rest of the lines are the sequence itself, split into lines of 80 characters. Here you can see part of the sequence of chromosome 13. But sequencing just produces a long string of nucleotides. How can you tell where each gene begins and ends? Annotating the human genome sequence posed another enormous challenge, which continues to this day. A tool called Gene Recognition and Assembly Internet Link, or GRAIL, was used to find the first genes. We can view genome annotations in GenBank format. The header information at the top contains information about the sequence itself, as well as the start and end positions of important features within the sequence. The sequence itself is listed at the end of the file. Here you can see the start and end positions of the BRCA2 gene, as well as the position of each exon. The file also contains the translated amino acid sequence for each coding region. You can view the genome in a more graphical way using the UCSC genome browser. Here you can see the BRCA2 gene, including the gene's location and orientation, which genes are around it, and what alternative transcript the genes code for. Another way to look at the genome is to use the NCBI Genome Data Viewer. Here we can easily zoom in to part of the sequence, select which tracks we want to show, and even set our own markers. You can also download the selected region in FASTA or GenBank format. One finding from the Human Genome Project is that individual genomes are 99.5% identical, which, put another way, means that a patient's genome differs from the reference genome at millions of sites. Most new variants are neutral and spread through the population through random processes. Variants are often shared by individuals with a common ancestor. For example, people with blue eyes are thought to have descended from a single individual who lived in Northern Europe 6,000 years ago. International HapMap Project. The Human Genome Project was only the first step and provided a haploid reference sequence that does not correspond to any actual person. Variants that occur at a frequency of at least 1% of the population are called single nucleotide polymorphisms, or SNPs. The International HapMap Project analyzed SNP frequencies in 270 people from 4 different populations, representing Africa, Japan, China, and Western Europe, in order to investigate patterns of genetic variation in humans. SNPs occur once about every 1,000 nucleotides, and each person has a distinct set of about 5 million SNPs out of approximately 100 million SNPs that have been cataloged so far. For simplicity, the human reference genome only contains 1 copy of each chromosome. But each cell has both a maternal and a paternal copy of each chromosome, and each one has a different set of SNPs. SNPs as genetic markers. Crossing over events during meiosis can generate novel SNP haplotypes. But the closer variants are to each other on a chromosome, the less likely they are to sort independently. This makes SNPs valuable as markers, even when the actual functional variant is unknown. And numerous genome-wide association studies have reported SNPs associated with specific diseases. For this reason, SNPs have been thoroughly studied, and a number of SNP resources are available, including dbSNP, the 1000 Genomes Project, and GnomeAD. Some SNPs are associated with increased risk of certain types of cancer. For example, RS3903072 is associated with reduced survival in breast cancer. SNP frequencies also vary by population. which can be helpful in filtering out common SNPs. Tomo's JMORP database includes whole genome data for 14,000 Japanese individuals. Even if we exclude common SNPs, this still leaves thousands of variants that are unique to each individual. Where do these variants come from? Some may be inherited as germline mutations and may be passed on to offspring. On the other hand, somatic mutations are those that occur during any cell division following fertilization and affect all progeny cells, but are usually not heritable. Mutation rates in humans are extremely low, about 1 mutation per 100 megabases. This is lower even than our closest relatives, chimpanzees. This is surprising since our DNA is constantly being damaged. The DNA in a single cell can be damaged up to 1 million times per day through environmental agents such as ultraviolet radiation, tobacco smoke, viruses, or oxygen-free radicals. DNA damage also occurs due to errors in DNA replication. DNA proofreading and repair mechanisms can quickly repair almost all damage, but some damage goes unrepaired. If the damage is not repaired quickly, the nucleotide change can become permanent. Even so, most mutations are harmless. Even within protein coding regions, the severity of a mutation depends on exactly where it occurs. Mutations at the third or wobble position in a codon often code for the same amino acid and represent a synonymous change. However, non-synonymous changes result in amino acid substitutions that might alter the structure or function of the protein. When a mutation introduces a novel TAA, TAG, or TGA codon, the protein sequence may be severely truncated. Insertion or deletion of nucleotides can also cause frame shifts that then code for completely different amino acid sequences. Genetic changes can also affect larger regions, including duplication or deletion of entire genes, resulting in copy number variants and structural variants. In some cases, a rearrangement can splice together parts of 2 different genes to create a fusion variant. Mutations that interfere with tumor suppressors, such as DNA repair or cell cycle checkpoint genes, can knock off the safeguards that protect genome integrity, unleashing a cascade of further mutations. Fortunately, since maternal and paternal copies of each gene are present, both copies must be disabled, either through mutation or by epigenetic silencing. This is the so-called Knudsen 2-hit hypothesis. On the other hand, a mutation that activates a single proto-oncogene can lead to cancer. For example, translocation of part of the ABL1 gene on chromosome 9 with the BCR gene on chromosome 22 creates a fusion gene that can induce chronic myeloid leukemia.